Welcome back to the lecture series on animal physiology in NPTEL section. So, today what we will be discussing is in the neuromuscular junction, how the energy is changed from electrical energy to mechanical energy. So, we talked about the neuromuscular circuit. Okay. So, here something is very interesting what you have to appreciate first before I draw the whole circuit and you know how the energy transduction is taking place. So, what exactly is happening is that, say for example, there is a signal, a sensory signal which is being sent by the sensory neurons to the spinal cord or maybe to the higher centers of the brain. Okay. From there, through the ventral route, a motor neuron is bringing the signal to the target organ, asking it, you move your hand like this or you walk like this, likewise XYZ, this could be any signal. So, once the signal comes, so signal is being transmitted as an electrical signal, electrical impulse, action potential, train of action potentials, ionic electricity is coming. But now that ionic electricity, eventually the end result what we see is that ionic electricity leads to the contraction and a movement of a muscle. How it does so? So, in other words, what is happening is that ionic electricity which is being transmitted by the nervous system is translated into some form of mechanical energy via a chemical route. So, here some molecules do that whole transduction, what we talk about the transduction procedure. So, there is a transduction which is taking place, electrical impulses through a chemical route translate into a mechanical energy, and that mechanical energy in the form of end output what you see is the contraction of a muscle. Okay. So, if you go back and you think about what is happening in the stretch reflex arc. So, in the stretch reflex arc what is happening? So, there is a stretch in your muscle. So, here is your muscle and there is a stretch in your muscle. So, there are stretch sensors all over it, what we call as intrafusal fiber or muscle spindle. So, they sense a mechanical change in them, a change in length, and then those sensors which are sitting here, they see the change, a mechanical change, and they translate that mechanical energy into electrical energy, first transduction. Okay. Then that electrical impulse in the form of ionic waves, ionic current waves travel all the way from here all the way to the spinal cord. Fine. After coming to the spinal cord, then there is a computation process taking place. That computation may be supported by the higher motor neuron which are present in the motor cortex of the brain, or maybe a very local computation which is taking place at the spinal cord level, especially in the borderline zone of the dorsal horn where the information enters, and the ventral horn, and through the interneurons in between. And those motor neurons which are sitting there then brings back the message, and all along this it is only the electrical impulses which are getting transmitted, the ionic electrical impulses. Okay. And then once it reaches the muscle at the neuromuscular junction, it transmits this electrical impulse to the muscle, and what the end result we see, there is a mechanical movement in the muscle accompanied by an action potential. Okay. So, how it does so now we will redraw the circuit, and after that there are three phases here. First phase is what I will do, I will redraw the circuit, neuromuscular junction, what I have just narrated you. Okay. Second thing is that we will look at the ultrastructure of the muscle, because that will help you people to appreciate that how the ionic current electricity is translated into a mechanical force via certain chemical routes. And third, we will club all these events together and we will appreciate this very wonderfully evolved phenomena. Okay. So, let us start with the section 5 and lecture 4, section 5, the nervous system. Actually, it's almost a part of the muscular system also, nervous system. Lecture 4. Okay. So, let us look at the circuit, how it looks like. So, here is the circuit element. This is the part of the spinal cord, and here you have the, in the blue zone in between is the ventral horn, VH, and this is dorsal horn, or the one which are carrying the sensory information, DH. So, motor neuron sitting here. This is the neuromuscular junction. Here is the target tissue, which is a muscle, and here is the sensory neuron, whose cell body is sitting slightly outside the dorsal horn, and here is the ascending pathway, and this is the local circuit where dorsal horn is synapsing on the ventral horn. Dorsal horn neuron is synapsing on the ventral horn neuron. And so, going by the arrow of information, so this is the arrow of information which is moving in the black. From here it is reaching this third level of computation, and it is coming back like this. So, today our objective will be to look at the cross section of this muscle in depth, in detail, because this is essential to understand this. Secondly, bit of a further detail of this zone. These are the two zones what we will be dealing today. So, one of the pertinent question which was asked sometime in 1960s was this. The question was like this. So, say for example, we have muscle like this, okay? It's a thick muscle, fine, or maybe some other part of the body where you have thick muscles. So, the neurons are coming all along like this and sending the signals. How the signal reaches all the way to the depth, because neuron is not really going all the way to the nitty-gritty detail. Why it kind of covered 2-3 layers down? Why the contraction? How that whole transmission taking place? So, if I have to draw, it will be something like this. Say for example, here is a muscle, here is your target muscle, okay, and here is a motor neuron which is coming, okay, and on the surface it is out here forming the neuromuscular junction. This is the cell body of the motor neuron, okay. So, here is your motor neuron, here is your target muscle. So, the governing question which made people to explore this is that how come the signal reaches all the way down? Because these are very small ionic electric current, and it has to travel all the way down. So, it may sound very simple, but it is actually a very big problem. So, what promotes this kind of transmission? So, there it all started. So, going back to the previous slide. So, now what we will do, I told you that there are two levels what I am going to discuss. So, this is the level 1, L1 I am putting it, level 1, and this is level 2. These are the two levels. So, first level is the one which I am now defining, that how this signal reaches all the way down. So, this is just I am showing like this. So, the arrow is pointing towards on the left hand side. Okay. So, here, so how this signal is moving. So, in order to understand this problem, you have to look in depth to the structure of the muscle. So, how the muscle structure looks. So, if I had to draw the muscle structure before that, and I am not drawing at this stage the ultrastructure. Ultrastructure is much more different. So, the structure of the muscle is something like this. So, if you look at muscle structure in the cross section, it is more like this. So, you see there are gorge kind of structures. Gorges are like, on two sides, if there are, think of it like if you have mountains like this, and there is a small narrow cliff, narrow gap in between, that is called a gorge. Okay. So, it is like a gorge like structures, and muscles are like that. So, coming back to the structure of the muscle, it is more like this. Because this structural feature has a big significance, that how the muscle conducts all these pieces of information. So, this is your skeletal muscle. Okay. Now, I told you, so this is in blue, what I am showing is wherever the neuromuscular junctions are getting formed. So for example, a neuron's processes are likewise, so on and so forth. Fine. So these are the zones of, these are the hot zones where you have all the neuromuscular junctions are getting formed, and these are where the neurotransmitters are getting released. Right, in the blue button like structure what I am drawing now, this is where. Okay. Now here comes our electrical impulse coming. Fine, it reaches here. So once it reaches here, what it does, it leads to the, these are the neurotransmitters. Let's in our situation we are talking about acetylcholine. So acetylcholine secretion leads to the opening of ion channels out here, the cation channels. So these cation channels leads to the entry of the sodium inside the muscle. Fine. After this, it is clear to all of you. So there is lot of cations which are entering. Entry of the cation channel, what it does, it leads to an action potential in the muscle. So if the muscle was sitting like this leads to an rise in the, so if it is sitting at minus 70 millivolt, this will lead to an action potential in a muscle like this. So now the muscle is experiencing, so if you have an electrode out here in a close proximity somewhere out here, if you have a, say for example, I place an electrode like this with respect to the A, and here you have a change in voltage. So you will be able to see a change in voltage like this. Okay. Now this electrical signal which is generated by the muscle because of the neuromuscular junction has a role to play. So now what is happening, this electrical stimulus from the neuronal side now transmitted into the muscle. So now this electrical impulse is moving on the muscle surface like this. Okay. And then maybe it will travel like this, and likewise it will travel. So while it is traveling, there is something very interesting happens. So the membrane is getting polarized, depolarized, polarized, depolarized, polarized, depolarized as this signal is moving all throughout till it dampens down. But while it is doing so, there is a local phenomena taking place at these junctures, because these are very specialized structure. So what I will do, I will highlight this part on this section. So out here within the muscle, I told you there are some specific organs called sarcoplasmic reticulum. They are all over present, mostly present like this out in this. These are called sarcoplasmic reticulum. Okay, SR, sarcoplasmic reticulum. Okay, the sarcoplasmic reticulums are the storehouse of calcium. They have lot of calcium in them, and it is very, very tightly regulated. So what happens when an impulse reaches here? So for example, this impulse reaches here. On the surface, as well as it's kind of not very clear, on the surface of the sarcoplasmic reticulum, whose membrane becomes continuous with the membrane of the muscle. So if I have to kind of again look at the finer details, so it is something like that. If this is the membrane of the sarcoplasmic reticulum, then this is the muscle membrane. They are almost kind of on top of each other. Okay, out here it is still not very clear how it is like kind of arranged. There is a series of voltage sensors sitting here, and at two level, at this. Let me use different color for you people to understand. So the one which I am now filling with green are the second level of voltage sensors. Okay, one of them are on the sarcoplasmic reticulum side, the other one is pretty much at the border of the sarcoplasmic reticulum and the muscle membrane likewise, which I am now. One of these sensors, again it is not clear till date one of these sensors changes a voltage change in the membrane, voltage change in the membrane, and as soon as it changes, senses the voltage change, it is kind of a hinge joint. It is, if I had to highlight this in much more in depth, it is something like this. This one is a sensor, and this one, as soon as this senses, it has a hook like structure like this, which is attached to the other sensor. So, as soon as this sensor, this one which is in the red, this one senses that there is a change in voltage, this sensor transmits a signal to this one, and this one raises up. And what exactly happens in that process is that this sensor, which is sensing in the completely red color. So, now there is a bit of a description what you have to kind of listen to me and process it slowly. So, say for example, I showed you the intersection zone of it. So, imagine this is one sensor, this is another sensor, my two hands like this. Okay. So, on my right hand side, this one. Okay. This one is the one which is sensing the voltage. So, there is a change in voltage. What it does, and this one is attached to the sarcoplasmic reticulum, because this is at the border. Once this senses, this asks it to open up like this, and as soon as this one opens, from sarcoplasmic reticulum, I told you, there is huge amount of calcium. The calcium goes out. So, what essentially happens at this stage is this, because if this receptor, the one which I was showing on with my right hand side, if this one gets activated, this one opens up a door out here, which opens up, lifts the cap, and then what you see that essentially immediate out here is a situation like this. And the series of molecules which are all over the place for a very transient period of time, and these molecules are Ca2+, calcium. What calcium does? And before I say what calcium does, let us talk about the nomenclature of these different receptors. So, here what you have is, now if I kind of show you in terms of in a much more bigger detail, it is, this is one which is opening up on top of the sarcoplasmic reticulum. Another gate is out here, which is in close proximity with it, which is the voltage sensor. And so, put the names now. Here is the voltage sensor, and here is calcium channel opener. So, this is sensing a voltage which asks the calcium channels to open. So, here is the voltage sensor, and this is asking it, come on, you have to open up. And as soon as this opens up, the next thing what we see along this whole milieu of the muscle is there is a lot of calcium which is coming out from the sarcoplasmic reticulum. Out of this, it has been discovered that this complex consists of two different protein which makes this voltage sensor and the calcium channel opener. One of them is called a ryanodine receptor, RYANO, DYNE, ryanodine, DYNE, ryanodine receptor. Sometime in short it is called RYR receptor. Okay. The second receptor is called dihydropyridine receptor, DYPR, or sorry, DHP, I think. That's how they pronounce it. Okay. these two receptors. So, where is the problem is that what is not clear is that which one is which one. So, they remain as a complex like this, something like a complex like this. If this one is the ryanodine receptor, then this one is the dihydropyridine receptor. So, what is not clear in this complex at the molecular details yet to mankind is which one is sensing voltage and which one is opening up the calcium channel. So, there is one which is actually opening up, or so there are two situations here. Some people believe that one of them is a voltage sensor, other one is the calcium channel opener. Fine, it acts like a hinge like this. Fine. There is another set of belief which says that both of them are doing both the jobs together. So, there is nothing like I will do this and I will do this, nothing of that sort. They do it together. So, this part is still a very hotly debated topic, especially this dihydropyridine receptor and ryanodine receptor has been extensively, very extensively studied in the cardiac system, especially in the cardiac excitability. Because one thing people have to realize, these are very programmable channels or programmable structures, because it not only sends out this calcium, it has a mechanism by which the sarcoplasmic reticulum pulls back the calcium. It is just like I take a bucket of water, drop the water in a room, and immediately within no time, within a delta t time, the water has flown. I have a mop, I just mop it out back to the bucket, from the bucket where I. So, they have a very tightly regulated mechanism. So, it is not like that, okay, I drop a bucket of water and then let the water kind of dries up, and it does not work like that. Biology is very well controlled system. It throws the bucket immediately in no time, pretty much in no time. By the time it has done its job, it is put back, because if it remain for a slightly excess moment, that will change the way our muscle twitch, the way signals are getting transmitted, that will be completely compromised. So, this is something what you people have to appreciate, that it is not that simple. It is all happening at a very narrow zone of time, within few picosecond, femtosecond, nanosecond, or at the max at a microsecond level. These things are all getting over. These molecular reactions are very well coordinated and very fast. So, as of now, it is not really clear out of this ryanodine receptor or dihydropyridine receptor which one is really doing the sensing job and which one is really opening up the channel. This is kind of still a hugely debated topic in the field of neuromuscular junction, muscle biology, cardiac physiology and all. Okay. Now, the next pertinent question comes, okay, there is a secretion of calcium, and then what we end result what we see, that there is a muscle contraction, but then what calcium does? How come calcium is essential for the muscle contraction to take place? So, as of now we are only talking about, so okay, there is a voltage sensing mechanism by which there is the all of us, like at that particular moment of time, the whole muscle milieu is all, there is a, if I have to plot the calcium wave, it will look like this, that in this diagram, yeah, something. So, what I will see essentially is that as soon as this signal reaches, so as soon as, if I have some way to plot it out here, the first signal is the neuromuscular junction signal. So, this is the neuromuscular junction, and the next thing, very soon I see that there is a wave of calcium. So, if I have the y axis telling calcium, so I will see a wave of calcium, a calcium wave going up, and this is the electrical signal. Okay, fine. So, this is, so what this calcium wave does, what this calcium spike, and I am showing spike because I told you that calcium does not remain there forever. As soon as it is being bathed, even faster than that, it is being pulled back by the sarcoplasmic reticulum. You cannot afford to have calcium sitting there and loitering around. Okay. So, then what this spike actually leads to, in order to understand and how this spike has some role to play in mechanical, electrical to mechanical transduction. That is what we are going to discuss in the part 2 of this, which is essentially this part L2. L1 we have understood now. Okay, this is what is happening. So, that is how a signal, when it reaches here through those gorge like structure, they reach all the way down, and then the electrical signals are reaching all the way down to the muscle. Now, we will be talking about how that is bringing about. So, this is how it is happening. So, it is reaching all the way, the muscle is receiving all the electrical signals all throughout like this. Now, we will talk about the structure of the muscle. So, as of now, what we have learnt about is, you have individual muscle cells. I have already discussed this part. Okay. These muscle cells are coming close to each other. These muscle cells are coming close to each other and forming the myotubes. These are the nucleus, these are the nucleus, these are the nucleus, these are the nucleus. Okay. So, these are the myocytes and these are the myotubes. What I essentially did not mention, so and eventually it forms myofibers and everything. Okay. What I essentially did not mention at that point of time is that this fiber, so these individual muscle cells have a crisscross of 2 different kind of proteins. I will come to that, what are those. Okay. So, all of them have like this. They are aligned like this in individual cells at different orientation, like this. So, when the muscle is attaching and forming a myotube muscle cells, so these proteins align themselves like this. And let me tell you, this is not on random, and we will come to that. These are the protein fibers which are constituting your muscle. So, what are these proteins? There are 2 proteins which are making this muscle structure here. The green one I represent as actin filament, and the blue one I represent as myosin filament. Here I wish to highlight something. This myosin, so actin fairly is well conserved. This myosin filaments have different variations, different variants. There is skeletal muscle myosin is different as compared to myosin of cardiac muscle, they are different. Myosin of smooth muscles are different. Myosin of intrafusal fibers are different. Intrafusal fibers. Okay. So, there are different variants of myosin. What does that mean? That means, in terms of functionality, they have different force generating abilities, and that essentially means force generating abilities. Okay. And now, if you look at it, so intrafusal fiber needs different kind of force, whereas skeletal muscle needs different kind of force, cardiac muscles need different kind of force, smooth muscle needs again another different kind of force. So, how this is being regulated? This is being regulated by this different kind of myosin fiber. But is it random? So, if during 1960s there is enormous amount of research which was done, it was figured out that this is not random. This is very, very well organized. So, how this is getting organized? So, I will have to do, these are the pictures what you see in the books, and they are kind of tough to figure out. So, I will draw a simple picture for you people, which will kind of clarify your doubts that how the muscle, how the cross section of the muscle. So, this is what we are doing. We are going through a cross section of a myotube or a myofiber, especially let us think of simple unit as the myotubes. Okay. So, let me see what is the color coding I am using. Okay, actin is in green, fine. I will keep actin as green. Take a bigger marker, fine. Now, actins are arranged. So, these are the green actins. Okay. So, now we are looking at the real cross section of a muscle. Okay. Now, on the other side, we have like this, like this, like this, like this, like this, like this. Fine. Now, we have another component which is a blue component, which is our myosin. Am I right? Yeah, fine. The myosin component, which is in blue. Fine. So, what I was trying to draw here, you see all these random patches out here. What you see here, they are not actually random. Now, I am looking through the microscopic structure. What you are seeing now. Okay. So, here you have the myosin filaments. So, the myosin filaments are not that simple as you think. These filaments have a head-like structure like this. We will come to the ultrastructural details of those. Likewise, okay. They have this kind of a structure. Then, yeah, like this. These are the myosin heads. We will talk about what those head means. Likewise. Okay. Then you have them like this. So, these are the structures which you will see in textbook, but they are a bit complex structure. That's why I am just taking that additional step to ensure that you people understand the whole biomechanics involved here. So, this is how the basic structure looks like. So, within this structure, this unit is called a sarcomere. Okay, this unit is called a sarcomere. Be very careful, because that is why I am drawing it in real time, so that you people understand this. Okay, this is called Z disc. And these are the terminologies which I can't help. You just have to, people have to kind of remember or appreciate it. And this zone is called the cross bridge zone. Cross bridge zone. Okay. And top of this particular part, which is under a microscope, these names. So for example, I draw a line like this, where you only see under a microscope the actin bands. You are not seeing the, once again. So, this is called, from here to here is called I band. This is a lighter band. Okay. Then from here to here, what is being called is called the A band. Okay, I band, A band, and let me give the color coding. This is your myosin, and this is your actin. Right. So, we finish all the color coding. This is very essential. So, now what I will do, I will come back to this drawing. Before I come back to this drawing, I will kind of give you an idea the ultrastructure, because I do not want to make it very crowded. Okay. Let us talk about the ultrastructure. So, what are our color codes again? Myosin. Let us talk about the myosin. So, while I was drawing the myosin, it looks like I was drawing it like this. Okay. I told you that there is a myosin head here. So, what I will do now, I will draw the okay. So, it is like this. So, this head has the ability to move like this. It can take a shape like this. Essentially, it can move like this. Okay. So, this this myosin head has a zone which is called with me, it is a complex drawing. So, this is called actin binding site. Okay. And there is just underneath it, there is something called a small zone out here. This is called ATP binding site, the energy molecule. Okay. ATP binding site. Fine. And there are some other nitty-gritty details which are not really essential for you people. These are called light meromyosin. You don't need to know this, but since if you come across in a book, kind of don't get confused. Okay. And out here you have the heavy meromyosin. These are different variants of myosin. Myosin. Okay. Meromyosin. Okay. And this is the tail of the myosin, which is dangling out there. Now, what we will do, we will draw the actin simultaneously. That will help you to kind of appreciate how the actins look like. So, here is the actin filament, which is in green. The situation, yes, actin is in green. Fine. So, actin has something called, this actin is something like, if you have to take a sectional view, it will be look like this. It will be more like a spiral kind of a structure of different proteins likewise. Okay. It looks like this, the way I am drawing it, different protein. Okay. So, if I represent the proteins like this, different proteins sitting out there. Now, if you look at this structure, the way it looks like is, within the actin you have something called troponin. There are proteins out here which are called, so you can represent them like, troponin, in accent troponin and tropomyosin, and underneath that you have the calcium binding site. This is very important because I told you that it is the calcium which leads to the mechanical energy transduction, from electrical to mechanical energy transduction. So, calcium binding site is somewhere out here. So, these are kind of, so imagine this is the calcium binding site, and on top of that is the troponin. So, what exactly happens in this situation? So, let us go back to the basic drawing again. So, what exactly is happening is this. Whenever, so look at this drawing. So, whenever under a normal condition, the contact between, okay, under normal condition they are not in touch. Fine, there is no touch. You look at this arrow, there is a gap out here. It physically they are not in touch. They are kind of close to each other, but they are not really bounded by anything. They are loose. Okay, they can move like this. Okay, this is not a problem. Okay, but as soon as the situation comes like this, calcium comes in. Now, this is the part what you have to understand. So, now there is a calcium influx because of it. So, the calcium comes and bind to the actins. Now, this is the calcium binding in red I am showing. Okay. As soon as the calcium binds there, it removes the blockage. So, the blockage is being created by, so the binding between actin and myosin cannot take place because it is the troponin and tropomyosin which blocks that. So for example, if my cell phone is here and if this is the myosin filament, then here is the actin, which is this hand, which is an actin. Actin has this tropomyosin, tropomyosin, all this cell phone. Okay. So, it is like this. Fine. So, now the calcium comes. As soon as the calcium comes, what happens? So, my mouth is on calcium now. So, in other words, what happens? The calcium comes, okay? Calcium removes this blockage like this. It's exactly something like that happens at the molecular level. The calcium comes and it removes the troponin tropomyosin blockage. So, as soon as that blockage is removed, the next thing what happens at this zone, if you look at in depth at this zone, what is happening at this zone, now the myosin head is in direct contact with the, here is the actin and here is the myosin head is in direct contact with the actin. Now, if you go back to the next thing, so here it has a ATP, so actin binding site. So, this actin binding site is now active. So, this is in touch with the myosin here, fine. Now, at this stage there is a ATP molecule which binds here. This ATP molecules helps to give it a, one second, oh shit. So, this ATP molecule coming back, okay. As soon as this is, so now think of, let us make this one more bigger, okay. Now, this is in contact and there is an ATP molecule which comes and binds here. Let me represent ATP with some other color, so that the confusion is not there. And this ATP molecules leads to a motion like this. And this motion, imagine now this motion is taking place in all of them like this. Essentially, these one can move other direction, this one will move in this direction, this one will move in this direction. So, all of a sudden it gives something called a power stroke. Okay, this is called a power stroke. When, and this power stroke is nothing but your, which leads to muscle contraction. Now, if you think of it, what exactly happened in this whole process? So, I told you that initially there is this nerve. So, let me go back to the slide. So, that will help you to appreciate now what happened in the first slide. Okay. So, here the electrical impulse came and this is transmitted through the ryanodine and sarcoplasmic, in the sarcoplasmic reticulum through the ryanodine and dihydropyridine receptors. And then there is a rise in calcium, and I showed you that calcium spike which took place. Okay. That calcium spike leads to a motion in the muscle, and that was essentially done because this calcium came because of the rise in calcium. This calcium which got rose up here, you see this calcium essentially went here and bind to the actin molecules, and after binding to them, and it has already, I showed you, it has an actin calcium binding domain out here within the actin molecule. It binds to the actin molecules and remove the blockage between the actin and the myosin. If this is the actin, this is myosin, there is a blockage because of some of the troponin tropomyosin. As soon as calcium comes here, it binds to it, and then this get, and it does a power stroke using ATP, and that is how the muscle contraction takes place. So, this is essentially is a electrical to mechanical energy transduction process, which is very, very fundamental all across the living system, and this is what I wish you people to appreciate. So, I will close in here. I hope you people have understood this concept. If you go through any picture online or book, now everything will make sense to you people. Okay. Thank you.